I am Hitans Kumar Shah from Centennial College in the course of Food Science Project 1 under the Professor Monica Datta. Today, I have taken one topic which is for my oral presentation, which is on the study of biofilm formation and antibiotic resistance pattern of bacteria that isolated from diabetic foot ulcer in St. George's Hospital in Democratic Republic of Congo. So I've taken this research article which was actually done by the Synco and team, and I'm taking this article and I'm going to present. I have presented details about that article and I'm going to represent this in front of you. So let me begin with that. So first of all, introduction is very important. So what is diabetes mellitus? So high level of blood sugar in our body where glucose cannot be utilized, which is called diabetes mellitus, and it can cause very different kind of disease. So the actual problem with antibiotic is antibiotic resistance, where bacteria cannot be killed by antibiotic. And food infection, if we talk about, then the food infection is a result from the biofilm producer and multi-drug resistance. And diabetic mellitus is also important here. So that all can affect here. So this study was conducted to determine the antibiotic resistance pattern of biofilm and diabetic foot ulcer that isolated from the patient. So it is very important to study about what is diabetic foot ulcer. So diabetic foot ulcer is a one kind of wound on the food that occurs in especially diabetic patient. So here it is very clearly visible in the image that what happens in every stage of diabetic foot ulcer. In stage 1 you see there is no defect. In the stage 2 you can see a red color smash or ulcer you can find on the stage 2. It is going to be more dangerous. You can find complete ulcer on stage 3, osteosis in stage 4, and in stage 5 you can see it's completely damaged the bottom part of the foot. So this is what happened in diabetic patient, and approximately 15% of diabetic patients are suffering from this disease, and 6% of patient has to get hospitalized just because of this infection. So it is also very important to know about what is biofilm formation and what is antibiotic resistance. So biofilm formation is something process where microorganism can irreversibly attach and grow on the surface and provide extracellular and produce polymers that facilitate attach to the matrix formation, where the antibiotic can make their wall very stronger so that the antibiotic cannot affect. And antibiotic resistance means bacteria cannot be killed by antibiotic. This image can easily shows that, like here for an example, this colonies, there is transparent growth in front of the colonies, which shows that antibiotic has effect against such kind of bacteria because it shows the zone of inhibition. While in this colony there is no such kind of growth, that means the bacteria is resistance against this antibiotic. This antibiotic cannot affect against such kind of bacteria. So this is what happened in all diseases. So we have to find out which bacteria are susceptible and which are resistant against in diabetic foot ulcer. Around 29.1 to 26.1 million people around worldwide are suffering from these diseases, and mostly patients are from the diabetes mellitus, because in diabetes, as you know, the immunity system are getting weaker. So there are chances of diabetes mellitus patient. 59.9 people are suffering from this disease that actually belongs to Africa only. Diabetes mellitus is condition where body cannot utilize the glucose normally and blood sugar level that goes up. So for that experiment we have used the method. So first method that we used here is origin of strain and laboratory procedure. Here we sterilize our saline and gauzes, and we took sample from the infected part from the patient in the hospital, and isolated bacteria we have taken on the TSA plate, trypticase soy agar medium, and we sent to the laboratory for the getting a data and doing a further experiment. The second is isolation and identification, where wound inoculated into the mannitol salt or McConkey agars, which are selective media, and we kept them in 37 degrees Celsius for 24 hours for in an incubation period. As aureus and staphylococcus both are like identified by the gram staining catalystase and coagulase. As you know, the catalyst both are gram positive, so catalyst test given whether they are positive or negative by the bubble forming reaction, and result were noted. Third is antibiotic susceptibility testing, which is very important test because here we used all kind of bacteria like erythromycin, azithromycin, gentamicin, ciprofloxacin, ofloxacin, and all susceptibility testing of this bacteria can happen. And we took sample from the gram negative strain also like gentamicin, amikacin, imipenem, trimethoprim, ampicillin. And after we kept all of the plates in the incubation for 37 degrees Celsius for 24 hours, and diameter of the zone that inhibited by the bacteria was also measured. And fourth is the biofilm formation. Like we all the screen all the isolated for their ability to form a crystal violet staining method, and McFarlane suspension with 0.5 turbidity was prepaid in TSB for each strain. And the interpretation of biofilm production, the average 3 walls has been calculated and adopted. So the result we were found is like that. Highest resistance we found in ofloxacin 83.3%, and trimethoprim and sulfamizazole and ciprofloxacin is around 75%. This strain was more susceptible to imipenem and vancomycin 91.7% and oxazoline 100%, and the other antibiotic shows resistance of 58.3%, which is average. So this result data can easily shows that like oxacillin and vancomycin were susceptibilities around 100% and 91.25% respectively. So which indicates that such kind of bacteria can affect against the biofilm producer and in diabetic foot ulcer. While you can see that erythromycin, methoprim, ciprofloxacin, ofloxacin, gentamicin, which are antibiotic resistance. That means that such kind of bacteria antibiotics cannot use in this such kind of diseases. Otherwise, it will not affect. Here there are more data for the Pseudomonas. We can see that only gentamicin is susceptible against Pseudomonas aeruginosa. Otherwise, all like ceftazimide, amikacin, imipedamine, ampicillin, ofloxacin, ciprofloxacin, all are resistant. So only gentamicin can be used in the treatment of such kind of bacterial diseases. Otherwise, there is no meaning of using any other antibiotic because bacteria produce antibiotic resistance. So in discussion, we can say that as DFU become a chronic disease and non-healing, despite having antibiotic treatment, the penetration administration antibiotic to site of infection is very uncertain, and it is effectiveness of such level is polymicrobial biofilm. Biofilm is very much responsible for that because it cannot able to affect antibiotics against the bacteria, and the result are like shows that the staphylococcus studies were resistant to mostly antibiotic tested, as we seen in the data table that mostly antibiotics were resistant. Bacteria were resistant against antibiotic. The bacterial infection generally is treated with oral drug. Antibiotics can be given orally. Is very rarely it happens that it can be given by IV or any other route. Generally it given in the oral route. So there are chances of infection. However, infection of chronic wounds are canny. Wounds can become infected by the bacteria that encapsulate themselves in biofilms over time, where body's natural defense mechanism is impaired. And biofilm is affected to delay in the healing of initiation of immune response in inflammatory cycle, which we seen in the result. So in the conclusion, we can say that the multi-drug resistant pathogen of DFU biofilm producer. So first of all, it is very important to understand the susceptibility and resistance of the bacteria, and afterwards it is better to give a treatment to the patient against the antibiotic because mostly bacteria are like antibiotic resistance. So it is better to study about that, and afterwards getting a treatment is better for the patient and even better for the doctor, for everyone. So this is what the aim of the study, that which are the susceptible and which are the resistant bacteria. These are the reference which I have using. I hope you like this presentation. Thank you so much for listening this. Thank you.